Hi, my name is Sierra Johnson, and for my final lab project, I'm going to be talking about the locomotion of three different phyla: phyla periphera, phyla nidaria, and phyla echinodermata. So, to get us started, what exactly is locomotion? Well, locomotion is the ability of cells or organisms to move and propel themselves from place to place. This function allows for organisms to do many different things, and evolution has enhanced organisms' success at these things over time. These include the ability to escape different predators, reproductive purposes such as being able to move to a certain area to reproduce or the ability to find mates, escaping from unsuitable habitats, and finding food and other resources. So the three phyla, like I mentioned, are going to be periphera, which are sponges, echinodermata, which are best represented by sea stars, and nidaria, which are hydra, including sea jellies, anemone, and corals. So let's start with periphera, our sponges. So sponges don't display tissue-level organization, but they do have specialized cells that perform specific functions. Now, most sponges in their adult stage are going to be completely sessile as adults, meaning that they're going to be attached to a substrate and stay there. However, in the larval stage, sponges will be motile, and they will use a flagella that they will push back and forth to be able to move in this larval stage. Now, while sponges don't show movement over large distances like other free-swimming marine individuals, there is some evidence that they're capable of creeping along substrates via organizational plasticity. Under some experimental conditions, researchers have shown that sponge cells spread on physical supports and can demonstrate directed movement. However, this is mostly not seen in the real world, and most sponges are completely sessile, meaning that they do not move from their attached space. Next, we're going to talk about nidaria, which are hydras. Now, these can be found in two different forms, polyps and medusas. Now, locomotion in polyps are typically seen to be sessile, with a single opening to the digestive system facing up with tentacles surrounding it. And these tentacles can be used, as shown in the diagram to the right, to give some movement to the polyp. However, most colonial polyps are going to be non-motile. Now, while they are typically sedentary, some colonies move slowly across substrates by actions of an inflatable peduncle, which is a stack that attaches to the strata in the lower end of the polyp. Sea anemones that are attached to substrates can creep slowly on their petal discs, or they can detach altogether, often in response to unfavorable physical conditions or attack by predators. However, again, most are typically sessile. Now, when it comes to medusas, which are going to be jellyfish and some other forms, are going to move through jet propulsion. This jet propulsion happens using ectodermal muscles, and the contraction of this circular muscle is going to squeeze the subumbular space, forcing out contained water, which is going to help the medusa move. However, most medusas, even with jet propulsion, aren't getting a lot of movement, so most of the time they're going to be carried by strong currents. Our last phylum is echinodermata, which are going to be represented by sea stars. Now, echinoderms possess a unique water vascular system consisting of a central ring canal and radial canals that extend along each arm. In this system, water circulates through the structures and facilitates gaseous exchange as well as nutrition, predation, and, of course, what we're discussing, locomotion. Now, the water in the vascular system is going to project from holes in the skeleton, and this is going to form the tube feet that you can see in the diagrams. These tube feet can expand or contract based on the volume of water present in the system of each arm. By using hydrostatic pressure, the animal can either protrude or retract their tube feet. Water enters the matoporite on the aboral side of the echinoderm. From there, it passes into the stone canal, which moves water into the ring canal. The ring canal connects to the radial canal, and the radial canals move water into the ampullae, which is each arm, and these tube feet are going to be where the water moves. By moving water through the water vascular system, echinoderms can move and force open mollusk shells during feeding. And the feet may be used as either levers by which these organisms step along a surface, or as an attachment for the animals to pull across surfaces. Either way, these tube feet are an incredible evolution by which echinodermata continue to move and function. Now, to compare the three phyla. As we discussed, echinodermata are going to move through their water vascular system. Nidaria are going to move through using tentacles or contracting their peduncles, and in medusas are going to be using jet propulsion or by currents. And lastly, periphera are mostly non-motile, which means that they're going to stay attached to a substrate, or in their larval stage, they'll use a flagellum to move. As you can see, going backwards through these different phyla, you can see how evolution has increased the locomotion of different organisms underwater. With periphera, we are completely non-motile except for in a larval stage, meaning that it is difficult to find ways in which you can move across different surfaces, and it is almost impossible to escape different predators or escape unfavorable habitats. With hydra, we move through currents in the medusa form, which is beneficial compared to the periphera. However, this largely allows a medusa to be very easily caught by predators, and the lack of movement means that it's difficult for them to escape unfavorable conditions in many situations. However, these medusa have other adaptations such as stinging cells to help with this lack of locomotion. And in polyps, we're mostly non-motile, which, being attached to a substrate, shows comparison to periphera. Lastly, echinodermata shows the farthest evolution from our non-motile and completely sessile periphera, being able to move across different surfaces to escape predators and unfavorable conditions.